Hello, it is I, the Mo. This is a video literally for Jada. So hi Jada. And honestly, you can probably skip this one because it's meiosis and it's really good background information, but we're gonna learn it later. And test on it later. But this is middle genetics in the unit 6. So heredity is all about shake what your mama gave you, and your mama gave you your DNA. So we want to talk really quickly about like just the source of genetic material. Well, it's the DNA. And remember from the previous unit, DNA is something that we can transcribe and translate to create proteins, which is the actual expression. So you'll hear the words, and you'll see genotypes, which is the DNA code. And then you'll hear phenotype, which is what it actually looks like. And just to remind you, bacteria, they have this bacterial chromosome, which is called a plasmid. It's circular DNA. It's still like a double-stranded, but it just is a circle. Versus more complex eukaryotes, they have linear DNA in these chromosomes. So overview, okay, to get to gene expression, big video here, having to move my face around, is the information content of DNA is in the form of specific sequences of nucleotides. And so you'll see a series of nucleotides in a certain order. If you're a eukaryote, you're going to go through mRNA modification. After you've transcribed that DNA code, you're going to get a specific set of mRNA that will code in codons to get you a protein. And that protein is going to be your actual expression. So these proteins are the link between the genotype. Proteins are specific per the DNA instruction, and the phenotype is their actual job and what they're doing. And that whole thing is gene expression, which we kind of talked about a ton already. So just to visualize that, DNA, multiple genes on DNA, and again, genes are just a sequence of DNA. And then we've got how we transcribe that, and we translate that into a specific polypeptide chain, which we will then fold and coil to create us a protein that will do a job, and that's our phenotype. So how do we get genes? And how do we like actually do this whole heredity thing? So we've learned how we like express that information. So now we just kind of have to understand how we get the information. So we don't necessarily inherit like the physical things. We've learned that we don't transform or we don't transfer proteins. So we're not actually inheriting any physical traits, but what we are inheriting is the genes that our parents have. And so those genes are the units of heredity. And they are passed down in the form of these things called gametes. So this video is all about meiosis, or it's about to be. And so we have to kind of understand meiosis to kind of understand why we are getting it. In the easiest form is you only get one chromosome from your mom and one chromosome for your dad, but there's a lot that goes into getting that one chromosome from each parent, because you have to do that 23 times to split the 46 chromosomes that a human has. Each gene on a chromosome has a very specific location, and that's its locus, and they match. So every parent has a set of chromosomes 1 through 23, and chromosome 1 in a dad has the same genes in the same locations as chromosome 1 in your mom and your kid and every single person. And so the chromosome structure is the same across everybody in that same species. And so that'll kind of take place when we get into evolution, how we talk about how certain species can't mate because their chromosomes are not the same. And that's one big piece of it, is that because their chromosome loci, their gene loci don't match. So they can't actually mate because the DNA doesn't talk the right way. And so DNA is typically packaged in these chromosomes. And like I said, one set of chromosomes is inherited from each of the parents. So I love this picture. It's a great picture. This is a chromosome. This is its matching chromosome. And so these are the two chromosomes that you inherit. The bands, these are the locations of the genes, and then on these genes are these things called alleles. And so a homologous pair of chromosome this is chromosome 1 as well as this. And everybody has 2 sets of chromosomes. So there are 2 alleles that you can represent because on each gene is an allele. And an allele is just the alternative form of that gene. Alleles cause the variations and they follow a bunch of different rules. One of the biggest rules is like the law of dominance. And so you've been learning this in 7th grade that if there is a dominant allele, which is typically represented by a capital letter, it will mask a recessive allele. And so when we talk about inheritance, you can only get one of these from each parent, and that's how you get 2 together to be able to read it. And so you get these chromosomes broken apart so that in sex cells or gametes, you're only putting one of every chromosome in it. And so that's that process of meiosis, where we have cells go through a process in which we're dividing them. But prior to dividing them, we actually replicated the DNA. So we go from we are 2 of every chromosome, where N represents the number of chromosomes. We replicate that and we're 4N, and then we divide that one time, we're back to 2N, and then we divide it one more time, and then we're just N. And so we call that haploid, in which you have 1 of every chromosome, which is 1N, versus diploid, which is what every other cell in your body is, where it's 2 of every chromosome. And that's called a somatic cell if it's your body cell and it has 46. So what will happen in meiosis is a female goes through meiosis and creates a haploid cell, an egg. A male goes through meiosis and they create a haploid cell, a sperm. And when those 2 things get together, they fertilize to create something called a zygote, which is diploid. And that's how many you're supposed to have. So that particular fertilized egg, now 46 chromosomes, able to, the DNA is able to talk to each other, is able to read correctly. That full count chromosome fertilized egg will now go through mitosis in somatic cells and create a human being. So really complicated process. We're going to zoom in on just a couple chromosomes so you can kind of see how it starts to happen. So we have 2 sister chromosomes of 1 replicated chromosome. So I want to make sure we're very clear right here. This guy right here and this guy right here are replicated already. So we're actually 4N in this particular thing, where we go, this is my dad's chromosome, this is my dad's chromosome, and they're the exact same. This is mom and this is mom. And so we've replicated them. These are homologous because these are both dad. They are held together by something called a centromere. Now, these are a homologous pair, but this is dad's and this is mom's, so these are non-sister chromatids. Sisters, non-sisters, even though these are the same chromosome, these are the same chromosome, and these are the same chromosome number. Okay, so we're very replicated. We're ready to go. We're going to start this process. I'm only looking at chromosome 1, 2, and 3. And we've paired them. They're floating around in different places, and then we've replicated them, and that's why we have 2. So chromosomes, the fancy way to say this is they're just supercoiled DNA, and the reason we want to organize that DNA is so that when we go through the process of meiosis, it's cleaner and it's easier to send to the next step. And again, homologous chromosomes, they carry the same genes that code for the same traits. You get 1 from your mom and you get 1 from your dad. And we're just saying that they are homologous in the sense that this chromosome pair and this chromosome pair are the same genes in the same location. So, more vocabulary. These chromosomes pairs have the same length. Again, they're held together by a centromere. When we replicate the parental chromosome, okay? And when we replicate the maternal chromosome. So we'll have these guys floating around. Right here, they are a diploid. Right here, it's actually a 4N. So when we have our diploid number of chromosomes, they have 2 sets of every chromosome. So there's one and there's one. And when we replicate DNA, it's going to look like this. So we've really got 1, 2, 3, 4. So a cell containing 2 sets of chromosomes 2N, one is inherited from each parent. That's a diploid cell. When we go through the process of DNA replication, we double that up so that we have a larger amount of DNA and we're ready to start our division. So haploid cells, reminding you, these are sex cells. This is an egg and a female and a sperm. Now they are only one of the chromosomes. So here was our pair. This is a diploid, and we got that from taking this haploid and putting them together. So for a human, somatic cells are diploid, and each of them have 46 chromosomes set into 23 pairs. But sex cells are haploid, and they are, single cells are haploid cells that have only 23 of the chromosomes. And we call meiosis reduction division. The reason we call it reduction division is because we are reducing the number of chromosomes. And it is a type of cellular division. So this is how we get sex cells. This is how organisms sexually reproduce. And meiosis takes place in sets of cell division called meiosis 1 and meiosis 2. But only one round of DNA replication occurs prior to the cell division, which is why we're able to divide twice and take a number 4N back to 2N, divide again, and we are a haploid cell. Each of the resulting cells are going to be haploid, and that's going to give us the ability to, pass on only one gene, which is all kind of the basis of heredity. Okay, so the stages of meiosis, again, we said we have meiosis 1. This is where the homologous chromosomes separate. This is considered the reduction division. It goes from one diploid, and then we're going to break it, and it's going to be 2 haploid daughter cells with replicated chromosomes. And so we've got the one of each, but now we've got to just divide it one more time. In the second division, it's the sister chromatids. They're going to separate so that it is a true haploid. This typically results in 4 cells, and this is called the equational division. So here's a picture of it. Here's one. Let's replicate. Let's divide. We got dad go over here with his homologous pair. Mom went over here with her homologous pair. We've got dad. I can pass down. Or mom. So I can pass down. There's a 50% chance I'm going to inherit the one that is my granddad's. Whoa, wild to think about. And 50% chance that I'm going to inherit my grandmom's. Okay? So how do we make sure that it is even further genetically diverse? Because this is how we get different siblings as well. There's this thing called crossing over that occurs during prophase of meiosis 1. This is an exchange of genetic material that gives new gene combinations. And the site of the exchange right here is called a chiasma. So how does this work? We have our 2 homologous chromosomes. And when we get ready to divide during prophase 1, they actually like high five each other. So what will happen is they are gonna touch and they will swap that genetic code so that the resulting cells, when we go through the set of division, again, dad's is over here, mom's is over here, we divide one more time. This individual haploid is all dad. This is a dad and a mom combo. This is a dad and a mom combo. And then this is just a mom. Okay, and so this is crossing over. This is how we continue genetic diversity, and we call these recombinant chromosomes. All right, I'm gonna end with just throwing this up here. You can pause the video if you want to read over it. This is different than the process of mitosis, so just wanted to compare. Peace.